You did everything right. You found a better spot. More light, better airflow, the perfect corner you'd been eyeing for weeks. You moved the plant carefully. You didn't damage the roots. You watered it when you got it settled. You stepped back and thought, there, that's better. And then the leaves started falling, not all at once, quietly, one or two at first, then more. Yellow, then brown, then just gone. Little circles of bare stem where leaves used to be. And the plant that looked healthy 3 days ago now looks like it's punishing you for trying to help. You moved it to a better place. It responded by falling apart. Most people interpret this one of two ways. Either they did something wrong they can't identify, or the plant is being difficult, ungrateful even. Neither explanation is satisfying, and neither is accurate. What your plant is doing when it drops leaves after being moved is one of the most psychologically fascinating stress responses in all of plant biology. It's not punishment. It's not randomness. It's a highly coordinated, metabolically expensive, evolutionarily ancient response to a specific kind of threat. The threat of the unknown. And when you understand what's actually happening inside those cells, you'll never interpret leaf drop the same way again. Let's build this from the ground up. The first thing you need to understand is that your plant didn't evolve to be moved. Not ever. In the wild, a plant germinates in a specific spot and lives there for its entire life. Sometimes decades, sometimes centuries. Its entire physiology is calibrated to that precise location. The angle of light, the humidity gradient, the temperature fluctuation between morning and night, the specific spectrum of wavelengths penetrating whatever canopy exists above it, the soil microbiome around its roots, the direction of prevailing air movement. All of these variables are not just tolerated by the plant, they are incorporated, encoded, used as reference data for every biological decision the plant makes. A 2018 study published in Plant, Cell and Environment by researchers at the University of Exeter examined how indoor plants calibrate their photosynthetic apparatus to stable light conditions over time. They found that after approximately 3 to 4 weeks in a consistent light environment, plants undergo a process called photoacclimation, a wholesale restructuring of the light harvesting complexes within chloroplasts to match the specific light spectrum and intensity of that location. More light in the blue spectrum? The plant shifts its chlorophyll ratios to absorb more blue. Stable low light? The plant increases chloroplast density per cell to maximize capture. Consistent directional light. The plant orients its chloroplast within each cell to optimize angle. This is not passive adaptation, it is active, energy-intensive physiological engineering, and it's calibrated to the exact light conditions of the exact spot where your plant has been sitting. When you move the plant, but even to a better spot with objectively more light, you invalidate all of that calibration in a single moment. The photosynthetic machinery that was precisely tuned for one set of conditions is now running in conditions it wasn't built for. And here's why that matters more than people realize. A study from the Max Planck Institute for Plant Biology, published in Current Biology in 2020, found that when plants are abruptly transferred from low light to high light conditions, even when both conditions are within the plant's survivable range, the sudden increase triggers photoinhibition. The light harvesting complexes, calibrated for lower intensity, absorb more energy than the photosynthetic system can process. The overflow generates reactive oxygen species, free radicals that damage chloroplast proteins and membrane lipids from the inside. The plant isn't thriving in the better light, it's being overwhelmed by it. And it needs time, sometimes weeks, to rebuild its photosynthetic machinery for the new conditions. So there's already a cellular crisis happening before the first leaf falls. But the leaf drop isn't caused by light adjustment alone. It's triggered by something deeper, something hormonal. Let me introduce you to the molecule at the center of this story. Ethylene. Ethylene is a gas, a simple hydrocarbon, 2 carbons, 4 hydrogens. And it is one of the most powerful signaling molecules in the plant kingdom. It regulates fruit ripening, flower senescence, root development. And critically, it's the primary molecular trigger for leaf abscission, the deliberate, controlled process by which a plant detaches its own leaves. A foundational study published in Plant Physiology by researchers at Cornell University first documented the relationship between environmental stress and ethylene production in the 1970s. Since then, the mechanism has been refined through hundreds of subsequent studies.
Welcome to the
The current model,
2021
summarized in a
Scientific
2021 review and annual
This
review
year,
of plant
our research
biology,
focused
works
heavily
like
on
this.
sustainable
When a
growth
plant encounters a
initiatives.
significant environmental disruption, change in light intensity, change in humidity, change in temperature, physical perturbation of the root system, it activates a stress cascade that includes rapid upregulation of ethylene biosynthesis. Ethylene concentrations in the plant's tissues rise, and ethylene acts on cells in the abscission zone, a thin layer of specialized cells at the base of each leaf petiole, where the leaf meets the stem. Ethylene triggers these cells to produce cell wall degrading enzymes, primarily cellulases and polygalacturonases, that dissolve the structural bonds holding the leaf to the plant. The leaf weakens at the connection point, and eventually it falls. Your plant isn't losing leaves because it's dying. It's cutting them off on purpose, deliberately, strategically. But why? Why would a plant voluntarily sacrifice healthy leaves in response to being moved to a better location? This is where the psychology becomes truly remarkable, and where we need to think about what the plant is actually calculating. When conditions change dramatically, a plant faces an immediate resource allocation problem. Its existing leaves were built for the old conditions. The specific waxy cuticle thickness, the stomatal density, the mesophyll cell organization, the chloroplast configuration, all of it is optimized for what was, not what is. Building new leaves calibrated for the new light intensity, humidity, and temperature, requires significant resources. And the old leaves, in their miscalibrated state, are simultaneously costing the plant resources to maintain while providing suboptimal photosynthetic returns. A study published in Functional Ecology by researchers at the University of Queensland in 2019 modeled this economic trade-off. They found that under rapidly changed conditions, the metabolic cost of maintaining and attempting to recalibrate existing leaves frequently exceeds the cost of dropping them and investing in new leaf development from scratch. Old leaves become stranded assets, and dropping them is not loss, it's liquidation. Your plant drops leaves after being moved, not because it's dying and not because it's confused. It drops them because it did the math. And the math said that shedding the old leaves and building new ones, calibrated to the new environment, is the most efficient path forward. This is strategic sacrifice, executed with hormonal precision. Now let's add another dimension. Because the ethylene story gets more interesting when you learn something about how ethylene spreads. Ethylene is a gas. It doesn't stay inside the plant. It diffuses outward from stressed tissue into the surrounding air. And neighboring plants, even of different species, detect ethylene and respond to it. A 2017 study published in the Journal of Experimental Botany confirmed that plants exposed to elevated atmospheric ethylene from a nearby stressed plant upregulate their own abscission signaling, even without being under direct stress themselves. The stressed plant broadcasts its distress chemistry, and neighboring plants interpret it as a warning signal, potentially triggering preemptive stress responses. Here's why this matters for the scenario of moving your plant. In an indoor environment, a small room, a shelf, a windowsill, if one plant is stressed and releasing ethylene, it may accelerate leaf drop in neighboring plants. You moved one plant, 3 started dropping leaves, and you had no idea they were talking to each other the entire time. But there's one more layer to this, and it's the one I think is most psychologically significant. Because the stress response your plant exhibits after being moved isn't just about light or humidity, a substantial part of it is about the root system. Roots are not passive anchors. They are the most sophisticated sensing organ a plant has. They detect moisture gradients, chemical gradients, gravity, mechanical resistance, temperature differences, and the chemical signatures of neighboring root systems. They are in constant, active communication with the soil microbiome, the specific community of bacteria and fungi that colonizes the rhizosphere, the zone of soil immediately surrounding the roots. A 2020 study published in Nature Microbiology documented that plants cultivated in a stable location develop highly specific, mutually beneficial relationships with particular microbial communities in their local soil. These relationships, particularly with mycorrhizal fungi, enhance nutrient uptake, provide chemical protection against pathogens, and may mediate plant-to-plant communication through high-fall networks. When you move a plant, even if you move it with all its original soil, you disrupt the continuity of that relationship. The root system encounters different soil contact patterns, different ambient microbial communities, different moisture distribution. The mycorrhizal network is severed, and without that network, the root's sensing and uptake capabilities are temporarily diminished. The plant is functioning blind, without its informational network, in an unfamiliar biochemical landscape. And it responds accordingly. A study from the Royal Botanic Gardens at Kew, published in 2019, measured root hair regrowth after transplant disruption and found minimum recovery windows of 2 to 4 weeks for fine root hair density to return to pre-move levels. During that window, water and nutrient uptake are significantly reduced, even if the plant looks intact from above. So here's the complete picture of what happens when your plant drops leaves after you move it. In the first hours, light conditions change. The photosynthetic apparatus, calibrated to the old location, is immediately operating out of spec. If light increased, photoinhibition may generate reactive oxygen species that begin damaging chloroplasts. Within hours to days, the stress cascade activates. Ethylene production rises. The abscission zones at the base of existing leaves receive ethylene signal. Cell wall degrading enzymes begin dissolving the structural bonds. The plant is already deciding which leaves to cut loose. Simultaneously, the root system is recalibrating. Fine root hairs disturbed by the move begin regrowing. The mycorrhizal relationships are interrupted. Water and nutrient uptake are temporarily reduced. The plant enters a period of genuine resource deficit even if it looks physically intact. And the plant's solution, the metabolically logical, evolutionarily selected response, is to reduce leaf area. Fewer leaves mean lower transpiration demand, lower maintenance cost, and lower water loss. The plant is right-sizing itself for the transition period, shedding what it can no longer efficiently support while it rebuilds the systems it needs to thrive in the new location. This is not failure. This is resilience architecture, built over millions of years of environmental instability. The leaves that fall are not the ones that died. They're the ones the plant chose to give up so that the rest could survive. Now here's what I want you to know practically, because understanding the psychology is only useful if it changes what you do. First, the drop almost always stops. Usually within 2 to 4 weeks, once the new photosynthetic calibration is underway and fine root hairs have regrown. If the drop continues beyond a month with no new growth emerging, then you have a different problem. But in most cases, the shedding is finite and self-limiting. Second, resist the urge to move it again. Every relocation resets the clock. Every move triggers a new ethylene cascade, a new round of abscission signaling, a new period of root recalibration. The worst thing you can do for a plant that's dropping leaves is move it back to its original spot. You've started the process. Let it complete. Third, reduce additional stressors during the transition. Don't repot during a move-related drop. Don't change the watering schedule dramatically. Don't add fertilizer, which can burn already stressed roots. The plant is managing one major transition. Give it the metabolic space to do it. And fourth, watch for the new leaves. Because they're coming. Calibrated to the new spot. Thicker or thinner, darker or lighter, built for exactly where the plant is now. Those new leaves are the proof that the strategy worked, that the sacrifice was worth it. And the next time you move a plant and watch the leaves begin to fall, don't see failure. See a plant doing the most intelligent thing it can do in an uncertain situation. Letting go of what it can no longer carry so it can build something new. If this changed how you see what happens when your plant loses leaves after being moved, subscribe. Because in the next video, we're going to explore something that sounds completely impossible. 7 strange things your plant can sense that you can't. And every single one of them is documented science. XX.